when you were sitting in that room, it sounds really fun, actually, like thinking about the peptides and things. Was there anything about the like peptide, the selection of the neoantigens and the peptides that were ended up being chosen? Anything that surprised you, about the ones that I know you did different peptide pools and anything that was a little bit not expected?
Yeah, it's a good question. I think the things that we thought about a lot of factors because there's a lot of opinions, but at the time, not a lot of data, not a lot of data to guide us how to really prioritize things. And so we had a particular scheme in mind. We looked at, the big picture stuff was the quality of the mutation. We want to make sure the mutation was real, it wasn't an artifact, and we want to make sure it was expressed at the RNA level because sometimes, especially these specific types of mutations like frame shift, the RNA can just be degraded very quickly, and so you might not actually lead to an antigen. You might not lead to a functional product. So we want to make sure it was at the RNA level. Um, we want to make sure it was a good predicted antigen. So we use a number of neoantigen prediction callers and really tried to prioritize ones that were predicted to be excellent antigens by both. And we had sort of a ranking scheme from there. And then from there, it got fuzzier because there were a lot of thoughts about what would be the best targets. Is there going to be mutations that are clonal? That sort of conceptually makes sense to me. They're going to be present in every tumor cell versus only a subpopulation. How much does the expression level, whether it's that much RNA or that much RNA or that much, is that really going to make a big difference? And then are they a driver, a passenger mutation? Are we crowding too much on one HLA allele type versus another? All of these sorts of things are things that we factored in as well. And we didn't know how it sort of, how would shake out essentially, which would be sort of be a priority. I think as we sort of looked back at the data, one of the things that was surprising to me is that it looked to be that some of these key driver mutations, so not just any mutation, but the drivers of kidney cancer specifically, those seem to have among the best immune responses that we observed, which I wouldn't have expected for both sort of technical and scientific reasons, but that's something that's sort of really interesting to us because, biologically, those are the ones we want to target. Those are probably dependencies of the cancer that are harder to sort of evolve their way out of. And so the idea that we might be able to target these key driver mutations is something that I'm certainly excited about moving forward.
Yeah, that was really neat. I wanted to ask a specific question on that. When I was reading through and seeing so many of the driver mutations represented in the peptide pools, and so VHL and BAP1, I think even PBRM1, but I think I noticed that maybe there wasn't much regarding SETD2. I didn't know if that was true or if there was a specific thought behind that.
Yeah. So it's a good question. And the real answer is a technical one, and this is where I think some of the challenges. I think peptides are a great platform for many reasons, but I think some of the practical challenges, you know, SETD2 was absent. Also, only I think we only had one patient that we vaccinated against their VHL mutation, where many more of those patients had VHL mutations, and a lot of those were predicted to be good antigens. But then when we actually go to manufacture the peptide, it's either, the character makeup, the amino acid makeup is such that it just couldn't be synthesized in adequate quantities, or it would precipitate out so that we'd put it into the peptide pool and it would crystallize and we had to essentially throw that peptide away. And so we definitely saw SETD2 mutations. It just after that process, unfortunately, none of them made it to the final sort of deliverable for a patient.
Okay, that makes sense. And again, kudos to the whole team and just all the amazing correlatives, that came out. Maybe if you would kind of walk us through, you've talked about the logistics and what went into the creation of the vaccine, and then tell us, how you basically studied if the vaccine worked for each individual and maybe some of the highlights of the science for us.
Yeah, absolutely. I think we had a kind of a number of key questions going into this. So some were honestly more practical. Is this feasible? Could we actually make this? The big sort of push in the past had been with melanoma, where there's a really sky high number of coding mutations. So lots of things to choose from, lots of neoantigens. Kidney cancer, much more modest. And so could we actually make this in a sort of widespread way? And so could we make it for all patients, for half of patients, for a third of patients? We didn't really know for sure going in. We had done some analysis that made us think we could, but we didn't know for sure. But that was the first question, and thankfully we're able to make it for all patients. The second is one of safety and tolerability, and I think for this peptide vaccination, that was typically okay. Kind of as expected, people got typically a day or two of feeling sort of flu-like symptoms, maybe from the vaccine, but also it was co-formulated with poly-ICLC, and so that certainly can cause that as well. But we saw that for essentially every patient, but no high-grade toxicities or anything like that. The third was broadly, is it doing what we're hoping it will do, which is steer the immune system or elicit an immune response? And that was really a big focus of our efforts. And so this was a huge number of essentially T cell assessments where we take their blood and isolate T cells or peripheral T cells before vaccination, during vaccination, after vaccination, and in a dish, add on the vaccine peptides and say, okay, are we seeing any reactivity to the vaccine? And when we started, prior to our vaccination, we essentially couldn't detect any in the peripheral blood. So they were either absent or at least below our limit, so low level they were below limit of detection. And then for every single patient, we saw that rise. We saw that we were able to generate reactive T cells for all patients. And then we could deconvolute and say which of the individual antigens was responsible for leading to that response. And that's where we found it was really a lot of these driver mutations. I think surprisingly, in some way, even though we're had predicted for CD8 antigens, CD8 T cell targets, the far majority of responses were CD4. And that's something that's actually been seen in other vaccine studies as well, including peptide based, but RNA based as well. And I think sort of has made me think a lot more than I have been about sort of the role of CD4 T cells in sort of anti-tumor immunity, certainly a helper role, but potentially direct anti-tumor roles as well. And so we talked about feasibility, and the answer was yes. Safety, yes. The next was really, is it leading to immune response? And the answer was yes there as well. The next was, is that really long lasting? Is this something where we get an immune response during vaccination and then it comes right back down? And so this was a little bit of a harder thing to track, but the way we did that is one, collecting these blood specimens throughout, but two, trying to identify which were the T cell populations that were most likely to be vaccine reactive. And we did that in a couple of ways. One is by just looking at the dynamics. So things that were basically absent or near absent initially and then increased by a certain threshold, we said those are likely to be vaccine reactive. But then we had a sort of an additional check on that, which was we did this delayed type hypersensitivity analysis, which is essentially, most people are probably familiar with a PPD that you do for tuberculosis, where you basically inject intradermally a small number of peptides related to tuberculosis. And if you've had immune response in the past, the T cells will rush in there and you'll form this little wheel. And so we did that for the vaccine peptides. We took all vaccine peptides, up to 20 of them, and at a low dose injected into the forum intradermally, waited 2 to 3 days to see if it formed a positive reaction. And for every patient, it did. Then we could biopsy that site and do single cell RNA and T cell receptor sequencing. So now we could say, okay, the T cells that rush to the vaccine peptides, almost certainly those are vaccine reactive. Otherwise they'd have no business being there. And so we could now track those specific T cell clones that were really vaccine reactive. And what we saw is they really went up with vaccination. They, dipped down a little bit, but then really persisted for months and in some case years after we had finished vaccination. So this was really a durable response. The next part was, okay, we vaccinated. Is that actually helping you attack tumor? And there's a colleague in the lab who always used to say, you could vaccinate against a piece of junk and you might be able to get an immune response against that junk, but that's not going to help you control the cancer. And so what we did was we tried to keep cryopreserved fresh tumor cells from each individual patient so we could then take their T cells after vaccination, expand them with a individual peptide, so the VHL peptide, the PBRN1 peptide, and then throw them back on top of that own patient's tumor to see if they would react. And it wasn't 100%, but in 7 of 9 patients, we saw that those T cells were then capable of reacting against tumor. And then the last part to look at is obviously, is there some sort of signal of clinical activity? And this is the hardest one, honestly, because this was a fairly high risk population, but it's only 9 patients. And so no one knows for sure these 9 patients that would have been cured by surgery alone. Again, we'd expect a third, maybe half to have recurred at a certain point, but it's 9 patients. So it's possible that just wouldn't have happened. we obviously were excited to see that none of those 9 patients had a cancer recurrence, but something we have to continue to follow over time. And the reality is we just have to test in a much larger number of patients.
Yeah. Well, that's a really great explanation. I think, walking us through the entire kind of scientific story and I think was, really, again, kudos for making the trial happen, for, showing the ability to do it, but then also just really being able to kind of learn from it, throughout the process and at the end.